Hello everyone, welcome to Get My Class. So let's solve this question which says, what is A and B? And they have given two pictures, that is A and B. We have to tell what are these. Option A is roots of control plant, B is transgenic plant roots 3 days after nematode infection, but got protection through RNAi treatment. Option B, A is roots of plant after nematode infection, and B is transgenic plant root 5 days after nematode infection, but got protection through RNAi treatment. Option C, A is roots of plant after nematode infection, B is transgenic plant root 3 days after nematode infection, but got protection through RNAi treatment. Options D, A is roots of transgenic plant after 2 days of RNA treatment, and B is roots of transgenic plant after 5 days of RNAi treatment. Now that's the question. Basically, we have to identify what is A and B. I know you all would have seen this picture in your book. So this is nothing but the roots of infected cotton plant. Okay. You can just say this is the picture of infected cotton roots. Okay. And this infection is caused by a parasite which is known as Meloidigyne incognitia. This is a type of what? This is a type of nematode. Okay. This is a type of nematode which causes infection in the tobacco plant. Now this one, what you see, this has been undergone the treatment. What kind of treatment? We call it as RNAi treatment. Okay. Or you can say it has undergone recombinant technology, or you can say transgenic technology, or it is a transgenic cotton plant. Transgenic cotton plant. Why transgenic cotton plant? Because we have performed a transgenic process where we have introduced the gene in the form of an RNA, okay, which produces an RNA in fact, and it will bring about the interference in the translation process, and it will convert the single-stranded RNA into double-stranded RNA. And that will, what will it do? It will silence that RNA and there will not be any production of the protein. So let me explain like this. So usually if you see the nematode I'm talking about, the pest, nematode for example, it causes disease in the cotton plant, right? Like in this particular example I'm talking about. So how does it cause disease? It has a gene for a particular protein, right? It has a gene for a particular toxic protein. So the gene will code for mRNA, okay? And the mRNA will give rise to protein. And this protein is what? This is what is the toxic protein, and this will cause the disease. That is what is happening here in the first case. Now, in the second case, what have been done to avoid this disease, we have stopped the production of the protein. Okay, and how did we stop the production of the protein? By silencing the mRNA of the nematode. How is that possible? So inside the plant cells, okay, what has been introduced? We have introduced the gene for the complementary mRNA of the nematode protein. Okay, so we have introduced what? The gene for complementary mRNA. So this mRNA, complementary mRNA by RNAi, this is what is called as RNAi method or I technology. Interference mRNA, this we call it as interference mRNA. Why? Because, so for example, if you have the, suppose this is the nematode mRNA, and we have introduced the gene for the complementary strand of this nematode mRNA, what will happen? This mRNA will form a double-stranded mRNA along with the nematode mRNA. So what will happen? This will become double-stranded and hence it will not undergo transcription or translation. They will not undergo translation and hence protein will not be formed. Okay, so that is what is RNAi method. Okay, and that has been done here in this particular plant. So this is a transgenic plant where RNAi treatment has been already done. That is why the root looked healthy. Okay, so let us see. Option A says the first one is roots of control plant, and that is option A. And B says it is a transgenic plant roots 3 days after nematode infection, but got protection through RNAi method. So that is what it says. But according to our explanation, what we wanted, Or what we said, the first plant is an infected plant. It is an infected plant. That's why you can see the roots like this. But here it says it's a control plant, so it is ruled out. Option B says the first one, that is A, is roots of plants after nematode infection. That is absolutely right. Then B is transgenic plant roots 5 days after nematode infection, but got protection through RNAi treatment. That is absolutely right. Okay, so option B should be the correct answer. Option C says A is roots of plants after nematode infection. That is correct. And B is transgenic plant root 3 days after nematode infection, but got protection through RNAi treatment. So it does take time. Within 3 days, you may not get the result. So it does take at least 5 days to show the result. Hence, option C is ruled out. Option D says A is roots of transgenic plant after 2 days of RNAi treatment. No, because if A is transgenic plant, it should have been healthy. But here it is infected. So here itself we can discard. So what is the correct answer? The correct answer is, let us conclude, that is option. The correct answer is option what? Option B. I hope you understood the explanation. Thank you everyone.